As a fisherman, I always knew that if you caught a fish and it was slow uh, to release when you did, you could always pull it back and forth, and it would help get the water over the gills to give them the little bit of extra oxygen that they needed to survive. But I never really ever thought about how the gills worked. So I decided that what better time than to look into that now and see how do gills actually work. This here is a picture of perhaps the biggest fish I've ever caught in my life. I don't know though. First of all, let's talk a little bit about the basic gill structure. You can see in this top left picture here that there's really three major parts uh, that make up the gills, which is the gill raker, which mostly is there to make sure that no food or any other kind of debris gets into the gill filaments, kind of pushes it away. Then there's the gill arch that provides the structure to the gill, and then the gill filament, which does the actual work of the gill that where the water flows by and the oxygen gets put onto the red blood cells. The way that it works kind of physically is either it's called buccal pumping, which is what most fish use, where when they open their mouths, their gill plates close. And then when they close their mouths, the gill plates open, and this rams water over the gills. And other than that, some other fish use, like sharks, we use ram, they'll like ram water just from swimming fast enough. And then it kind of works, the heart will pump blood directly to the gills, and then it'll come all the way through all the capillaries in the body, get oxygen wherever it needs to go. So that's a little bit of a basic explanation of how they work. So now we can get a little bit more technical. So lamellae are protrusions off of the gill filament, which is the part that I said did the actual work. And that increases the surface area. So more surface area means that there's more place for the oxygen to get pulled out of the water and onto the gill surface, where it can be put into the blood. And if you look on this picture here, it's got all these little holes, and this is a picture of a gill filament with a lamellae on it. And these asterisks here show where the blood flows through them. And so you can kind of get a good idea of how much surface area is really being used in order to maximize the amount of blood that can, or the amount of oxygen that can get pulled out of the water. So this is a scanning electron micrograph of an American stingray gill filament. Also, the active sites of gill filaments have the two types of cells. There's the pavement cells and then the mitochondria-rich cells. And the mitochondria-rich cells are really the ones that do a lot of the work. People consider them to be the active sites. And so we'll talk a little bit more about that here. So they're also called chloride cells, and they're found in gills, the larval epidermis, and brood pouches of pipefish, which pipefish are very similar to seahorses. They keep the fathers to hold the eggs in a brood pouch until they are ready to kind of come out and they hatch. So the primary purpose of these mitochondria-rich cells is they transfer excess ions out of the body. So you can see in these pictures here that there's all these black dots on this bottom picture are mitochondria-rich cells. So you can see that there's a lot of them right around the gills of them. And you can also see in this top picture, all these little holes are all filled with mitochondria-rich cells. But that makes me think, everyone always says that gills are for respiration, but that indicates if they're set up for ion transfer, that indicates that they don't just do respiration. So there is a study about the pirarucu, which is a Brazilian species of fish that breathes up to like 70%, 75% air. And that grows as it develops. So smaller fish have a more developed gill system, and then as they grow and start breathing more air, they need it less. And so this study was looking into this here, and you can see in this graph that a lot of what they do, especially on the ammonia excretion, is very, like, the small fish excrete a significantly amount more ammonia than the large fish. And that's just because the gills are so much more developed in the younger fish that they have the ability to get rid of that ammonia, where the larger fish don't. You can also see on this ion flux, seems like the only two ones that are significant are sodium and potassium, which are both significant in favor of the larger fish, but that can also be explained by just the fact that it's so much larger. And I think there might also be, like we saw, that the pipefish in the last slide had the mitochondria-rich cells all over its body, not just exclusively on its gills, but I think that had plays a big part there. But up to this point, we've mostly been talking about gills and fish, and as we know, there's like not only fish have gills. So this picture here is of an axolotl, which I kind of want an axolotl now after doing this project. They're pretty cool. And I guess people keep them as pets. But most amphibian larvae will breathe through their skin and through gills. And frog tadpoles can also use their large tails, but their gills kind of operate in the same way, but they have a little bit different structure, like especially with axolotls and a lot of other salamanders, they never get like develop the gills. They keep them throughout their whole lives.